Good day, everyone. So welcome to my YouTube channel. I'll be discussing gelatin hydrolysis test, which is an important biochemical test in the identification of an unknown organism. Gelatin hydrolysis test or gelatin liquefaction test is an important test in the identification of bacteria. Gelatin is present in partial digest of animal protein. There are two methods in the gelatin hydrolysis test. This includes the first one here, nutrient gelatin stab method. It's a standard and most commonly employed method. And here, the nutrient gelatin tube is inoculated with the test organism by stabbing into the bud, and the tube is incubated at 37 degrees Celsius for 24 hours. After that period of incubation, the tube is transferred into a refrigerator and removed after 30 minutes to 1 hour and examined for liquefaction or gelatin hydrolysis. If there is no liquefaction, the tube is returned to the incubator for another 24 hours and then checked for liquefaction again after being placed in the refrigerator. This is done for 7 days. Here is a plate showing a positive gelatin liquefaction test. Notice in the upper part of the test tube, gelatin has been liquefied or hydrolyzed. This is because the test organism has produced the enzyme gelatinase, which hydrolyzed or liquefied the gelatin present in the tube. While plate 2 shows negative gelatin liquefaction test, so there is no evidence of liquefaction of gelatin or hydrolysis of gelatin. The second method is the nutrient gelatin plate method. A heavy inoculum of a 24-hour test organism is tab inoculated on culture plates pre-filled with nutrient gelatin and then incubated at 37 degrees Celsius for 24 hours. If the test organism produces gelatinase, evidence of gelatin hydrolysis is indicated by clear zones around the gelatinase positive colonies following flooding of the gelatin plates with acidic mercury chloride. In some cases, plates are flooded with saturated ammonium sulfate to precipitate the unhydrolyzed gelatin, making the clear zones easier to see. Results are often observed within 5 to 10 minutes after flooding with saturated ammonium sulfate. Here is a plate showing the nutrient gelatin plate method. Isolate A here has a clear area around it, showing evidence of gelatin liquefaction or gelatin hydrolysis. That is, the isolate has produced gelatinase, which diffuse into the medium and hydrolyze or the gelatin. while isolate B, there is no clear zone of hydrolysis of the gelatin. What's the application of gelatin hydrolysis test? The ability of bacteria to produce gelatinase is detected by the gelatinase hydrolysis test. It distinguishes the gelatinase positive pathogenic Staphylococcus aureus from the gelatinase negative non-pathogenic Staphylococcus epidermidis. The test also aids in the identification of other bacteria genera, which include Serratia, Pseudomonas, Flavobacterium, and Clostridium. Gelatinases are proteases secreted extracellularly by some bacteria, which hydrolyze or digest gelatin. Here are some references and links for further reading. Thank you for listening.